So, good evening, everyone. I am Dr. Satyabrata, MDDM clinical pharmacologist, and today I am going to discuss on dynamics and modes of disease transmission. So, coming to dynamics of disease transmission, the chain of infection includes susceptible host, modes of transmission, and source, that is reservoir. And communicable diseases are transmitted from the reservoir or source of infection to susceptible host. And there are three links in chain of transmission, that is reservoir or source, modes of transmission, and susceptible host. Coming to the sources and reservoir. Source of infection is defined as the person, animal, object, or substance from which an infectious agent passes to the host. And reservoir is defined as any person, animal, arthropod, plant, soil, or substance in which an infectious agent lives and multiplies, on which it depends primarily for survival and reproduces to transmit to a susceptible host. For example, in hookworm infection, reservoir is man and the source is soil contaminated with infective larvae. Then, what is homologous reservoir? What is heterologous reservoir? Homologous reservoir is from the same species, that is human to human. For example, man infected with Vibrio cholerae infection, that is cholera. And heterologous reservoir from animal to human. For example, from animal or bird or poultry infected with salmonella. Then coming to types of reservoir. Human reservoir, animal and non-living things. So, human reservoir. The case is that a person in population or study group identified as having the particular disease, health disorder or condition under investigation. Types of cases. Clinical case, clinically confirmed cases. Subclinical case. The disease agent may multiply in the host but does not manifest itself by signs and symptoms. For example, mumps, polio, hepatitis A, hepatitis B. And what is latent case? Latent case means host does not shed infectious agent which lies dormant within the host without symptoms. For example, herpes simplex. Then important epidemiological terminologies, that is primary case. Primary case refers to first case of a communicable disease introduced into the population unit being studied. Index case, first case to come to the attention of the investigator. It is not always the primary case. Then secondary case, those developing from contact with primary case. Then suspect case, an individual or group of individuals who has all of the signs and symptoms of a disease yet has not been diagnosed as having the disease. Then coming to carriers. An infected person or animal that harbors a specific infectious agent in the absence of clinical disease and serve as potential source of infection to others. According to types, incubatory carriers, that is during incubation period before clinical illness begins. For example, measles and mumps. Convalescent carriers, that occurs after recovery from illness. For example, typhoid, dysentery. Healthy carriers harbor subclinical infection and shed disease agent. They emerge from subclinical cases. However, subclinical cases may or may not be act as carriers. For example, polio, cholera. According to duration, carrier is classified further into temporary carrier. That means carrier sheds agent for short period. Chronic carrier sheds agent for indefinite period. For example, hepatitis B and typhoid are chronic carrier. Then according to portal of exit, carriers further divided: urinary carriers, intestinal carriers, respiratory carriers. For example, typhoid, urinary carriers more dangerous than intestinal, food establishment more dangerous than office establishment. Animal reservoir, for example, zoonotic diseases, rabies, Japanese encephalitis. Non-living things reservoir, for example, soil is reservoir for in case of tetanus, anthrax, mycetoma. Then coming to modes of transmission of disease. There are different types of modes of transmission of disease. One is direct transmission, other is indirect transmission. In direct transmission, the disease can be transmitted either direct contact or droplet infection or contact with the soil or inoculation into skin or mucosa, and transplacental or vertical transmission. And in case of indirect transmission, that can be vehicle borne transmission, vector borne. Vector borne is further mechanical and biological transmission. Airborne is for the droplet nuclei and dust. And then fomite borne transmission, then unclean hands and fingers. These are indirect transmission. Then coming to direct transmission, direct contact from skin to skin, mucosa to mucosa, or mucosa to skin. For example, leprosy, acquired immune deficiency, AIDS, STD, sexual transmitted diseases. And then coming to droplet infection. Droplet infection by direct projection of droplets of saliva and nasopharyngeal secretions during sneezing or coughing. For example, common cold, COVID-19, tuberculosis. Then contact with soil. Contact with soil, for example, hookworm larva, tetanus. And inoculation into skin oblique mucosa. For example, rabies virus by dog bite, hepatitis B virus through contaminated needles and syringes. These are transmitted through inoculation into skin oblique mucosa. And then another mode of transmission is transplacental oblique vertical transmission. That is, for example, torch agents, hepatitis B, acquired immune deficiency AIDS, and thalidomide drug. So torch agents. These are transplacental oblique vertical transmission. Then coming to indirect transmission. Indirect transmission by 5 F's, that is flies, fingers, fomites, food and fluid. One is vehicle borne transmission, that is transmission of agent through water, food, blood, etc. For example, hepatitis A, typhoid, hepatitis B, syphilis. Then vector borne diseases, an arthropod or any living carrier that transports agent to a susceptible individual. Epidemiological classification of vector borne diseases. By vector, invertebrate types, that is diphtheria, flies and mosquitoes are included, and Cynoptera, that is fleas, Orthoptera, it includes cockroaches, Anoplura includes lice, Hemiptera includes bugs, Acarina includes ticks, mites, Copepoda includes cyclops. Then come to vertebrate type. Vertebrate type includes mice, rodents, bats. By transmission chain, man and a non-vertebrate host. Man, arthropod man, that is malaria. Man, snail man, that is schistosomiasis. Man, non-vertebrate host and vertebrate host. Mammal, arthropod man, for example, plague. From bird, arthropod to man, that is Japanese encephalitis. It is transmitted by chain. Man and two intermediate hosts, that is man, cyclops, fish, man. In case of fish, tapeworm. From man to snail to fish to man, Clonorchis sinensis, that is liver flu, can be transmitted like this. From man to snail to crab to man, that is paragoniasis, that is long flu. It transmitted like this. This is a chain of transmission. Then by methods in which vectors transmit agent, that is biting, regurgitation, plague by rat fleas, agent Yersinia pestis, blocks proventricular valve of rat flea. Then scratching is another method with body fluids of vectors. By methods in which parasites transit and propagate through vectors, mechanical transmission, there is no multiplication of infectious agent but mechanically transported by vectors, for example via proboscis or GIT gastrointestinal tract or excreta. Then types of biological transmission. Propagative, the agent merely multiplies in vector but no change in form. For example, plague bacilli in rat flea, that is propagative transmission. Cyclopropagative transmission, agent changes in form and number in this type of transmission. For example, malaria parasite in mosquito. Then cyclodevelopmental transmission, agent undergoes only development but no multiplication. For example, microfilaria in mosquito. Then airborne droplet nuclei by evaporation of droplets and aerosols, for example COVID-19, influenza, chickenpox, measles. And dust transmission, that is in case of pneumonia, for example pneumonia, Q fever, C. borneti, and tuberculosis. Then fomite-borne, that is substances other than water and food like clothes, towels, books, glasses, taps, etc. For example diphtheria, typhoid, dysentery, hepatitis A. These are fomite-borne transmission. Then unclean hands and fingers, staphylococcal infection, typhoid, dysentery. These are transmitted through unclean hands and fingers. Then coming to susceptible host, a person lacking effective resistance to a particular pathogenic agent. 4 stages are required for successful parasitism, that is portal of entry, site of election inside the body, portal of exit, and survival in external environment. So susceptible host with chain of infection. Susceptible host, infectious agent, reservoirs, portal of exit, means of transmission, portal of entry. So example of susceptible host, immunocompromised, elderly, chronically ill, these are susceptible hosts. And periods of infectious diseases, that is incubation period, time interval between invasion by an infectious agent and appearance of the first sign or symptom of the disease. Then median incubation period, time required for 50% of the cases to occur following exposure. Then latent period from disease initiation to detection, usually more applicable for non-infectious diseases. Then periods of infectious disease, incubation period, latent period of communicability, and you can go through different books and articles to learn more about these periods of infectious disease. And then this picture shows showing incubation periods, exposure, primary cases, secondary cases, and you can go through different books to learn more about this. Then uses of incubation period, tracing the source of infection and contacts, determining the period of surveillance, usually equal to maximum incubation period, and applying immunization principles helps to prevent clinical illness by active or passive immunizations. Identification of point source or propagated epidemics. Point source epidemic, that is all cases occur within one incubation period. Propagated epidemic, that is cases occur later than known length of incubation period. Estimating prognosis, that is poor prognosis in diseases with shorter incubation period, IP. For example, tetanus, rabies. Then what is secondary attack rate, SAR? SAR secondary attack rate is defined as number of exposed persons developing the disease within the range of the incubation period. SAR is equal to number of exposed persons developing the disease within the range of the incubation period divided by total number of exposed or susceptible contacts into 100. What are the uses of secondary attack rate? Uses. It is used to determine whether a disease of unknown etiology is communicable or not, and it evaluates the effectiveness of control measures such as isolation and immunization. Then host defenses. Resistance exhibited by the host against any foreign antigen. And then what is acquired immunity? Acquired immunity that develops during your lifetime. It can be active immunity and passive immunity. Active immunity develops in response to an infection or vaccination. And active immunity can be further natural and artificial. Natural means antibody developed in response to an infection, and artificial means antibodies developed in response to a vaccination. Then coming to passive immunity develops after you receive antibodies from someone or somewhere else. It is also natural and divided into natural and artificial. Natural passive immunity means antibodies received from mother, for example through breast milk, and artificial passive immunity means antibodies received from a medicine, for example from a gamma globulin injection or infusion. Then coming to active immunity acquired in 3 ways. Following clinical infection, for example measles, chickenpox, rubella, and following subclinical or inapparent infection, that is for example polio and diphtheria. And another is following immunization with an antigen, for example killed vaccine, live vaccine, toxoid. So the immune response, what are the characteristics of immune response? Primary response, secondary response. Exposure to antigen is the characteristic. Primary response is fast exposure to a specific antigen, and secondary response is after second exposure to same antigen. Primary response, time of onset, in case of primary response 1 week delay, and in case of secondary response within hours. Strength, primary response weak potency, secondary response more potent. Duration, primary response short life for only a few weeks, secondary response forms antibodies for many months. Primary response predominant antibody IgM more than equal to IgG, secondary response IgG more than IgA, IgE, IgM. Memory cells, in case of primary response no memory cells, but secondary response there is, yes, memory cells are present. Then primary versus secondary antibody responses. This is the graph showing the same. Primary, in case of primary response there is a lag period. On 5 days there will be IgM, total antibody will increase up to 15 days. Then IgG will be increased after 15 days, then decline, and total antibody will plateau on 15 days, then decrease. In case of secondary response, after primary response, on 10 days the IgM will peak, and there will be total antibody will be increased, and IgG will be peak on 15 days, then decline after 15 days. So x-axis showing antibody concentration in serum, and y-axis is days or time. Coming to humoral and cell mediated immunity. And in case of humoral, intact antigens stimulate the B cell, plasma cells and memory B cells are produced, and antigens engulfed and displayed by phagocytes, TH cell will stimulate the B cell, Then plasma cells and memory cell B cells will be produced. Memory TH cells will be produced. And humoral, it is secret antibodies that defend against extracellular pathogens. In case of cell-mediated immunity, antigens displayed by infected cells, TC cells activate, and memory TC cells and active TC cells will be formed. And in case of cell-mediated immunity, defend against infected cells, cancers, and transplant tissues. And the humoral mediated TH cells activate the TC cells in cell mediated. And also TH cells will inhibit TH cells or activate TH cells. This is a complex method, and you have to learn more and more books and articles about humoral and cell mediated immunity to learn more about this topic. So please go through different books to learn more about this humoral and cell mediated response or immunity. Then coming to passive immunity, it can be induced by administration of ready-made antiserum or immunoglobulin, transfer of maternal antibody across placenta, transfer of lymphocytes to induce passive cellular immunity. Still, it is experimental. Then difference between active immunity and passive immunity. In case of active immunity, mediated by the antibodies produced by the person's own cells in response to direct contact of an antigen, and it does not generate a rapid response. It lasts for a long time, and active immunity more effective protection, and secondary response is more enhanced, and it does not work in immunodeficient hosts. But in case of passive immunity which results from introduction of antibodies from outside, passive immunity generates a rapid response. It may not last for long. It is less effective. No immunological memory occurs in case of passive immunity, and it works in immunodeficient hosts. Then coming to herd immunity, that is community immunity. It is a type of immunity that occurs when vaccination of a portion of population herd provides protection to unprotected individuals. The higher the number of immune individuals, the lower the contact of susceptible host with infectious agent. It provides immunological barrier to the spread of disease in human, and elements which contribute to herd immunity are occurrence of clinical and subclinical infection in the herd, immunization of herd, and herd structure that is never constant. Then by immunization program, susceptible persons are reduced to a small proportion, leads to elimination of the disease, for example diphtheria, poliomyelitis. Herd immunity threshold, the proportion of immune individuals in a population above which a disease may no longer persist. Its value varies with virulence of the disease, efficacy of the vaccine, and contact parameter for population. So that's all for today. You can go through different books, different articles published on this topic of passive immunity, herd immunity, etc. to learn more and more about this. Please like and subscribe my channel, and also watch complete till the end of these videos, and so that you can learn a lots of things of different diseases and their management, and also different other topics. So please watch more and more, like and subscribe, and that's all for today. Thank you very much. Your dedication to infection prevention helps save lives. So thank you very much. Keep watching. Jai Jagannath.